Hello students, welcome back to the biology class. So in the last class we learned that the transcription is very much different from the replication. We learned 4 points of differences. We also understood the enzymes involved in the replication, how they are different in their functions in the case of the prokaryotic replication machinery and the eukaryotic replication machinery. We even understood that the two reasons of why that during transcription only one strand of DNA is read, because in replication both undergo reading. And we understood the two reasons. The first reason is though the both strands of DNA are complementary, they are not identical to each other. So when they code for the RNA formation, the RNA formed will have different reading of their codons. And because codons are different, the amino acid brought would be different. So though the two strands would be complementary to each other, they will code for formation of different proteins, thus confusing the machinery of the transcription and translation. And second reason is, if both strands undergo copies, then what will happen? Both strands of the RNA, being complementary, may come close to each other, develop hydrogen bonds, and this will cause formation or development of a double-stranded RNA, and such an RNA will not be functional or reactive, so it won't participate in the process of the translation, because during translation a reactive RNA, that is always a single-strand RNA, has to join with the when we say RNA, I mean messenger RNA, has to join with the ribosomal rRNA or ribosomal RNA of the small subunit of ribosome. Then only translation or protein formation can start. So this much we did in the last class. Let's understand now the next topic, and that is the transcription unit. A transcription unit is the DNA which is defined in a DNA is defined as primarily by the three regions in DNA. So in the last class I told you, during replication full length of DNA is read, but during transcription only a segment of DNA would be read, and that segment can also be called as transcription unit, or it can also be called as a length between or distance between the promoter and terminator, or you can say what is read basically is the structural gene. So if we draw a diagram in this manner, please refer NCERT side by side to understand these concepts better. There is a convention in defining the two strands of DNA in the structural gene of transcription unit. Since the two strands have opposite polarity and the DNA dependent RNA polymerase enzyme will also catalyze in only one direction, that is the 5' to 3'. Even in the last class we learned that whenever DNA is read, it will be read in 3 to 5 direction, and addition would be in 5' to 3' direction. The strand which has the polarity 3 to 5 will act like template. So in this case, because promoter is here, so starting will be from here, and this will be the ending. So promoter is here, so the enzyme will come and attach to the promoter. The enzyme name is DNA dependent RNA polymerase enzyme. Now there are two strands. Which strand should the enzyme read? So the reading is always from the 3'. So this strand will act like a template strand and will cause the formation of RNA in 5' to 3' direction. So this strand is called as the template strand, and the other strand which has the polarity of 5' to 3' and the sequence same like RNA because they are complementary. So when this strand of DNA will cause the formation of a RNA, the reading of the coding strand of DNA will be almost similar to RNA except T and U. Wherever there is T here on the coding strand, it will be U on the RNA. The other strand with the polarity 5' to 3' in the sequence same as the RNA except thymine at the place of the uracil is displaced during transcription. And this strand which does not code for anything is strangely called in biology as the coding strand. So coding strand reading is same like the messenger RNA. That's why it is also called as sense strand. And this one is called as the opposite, so it is also called as the anti-sense strand. These names are missing in NCERT, but they are given in your help books as well as your MCQs books also. now there is a fixed rule in biology, in genetics, that all reference point while defining this topic is made with the help of the coding strand. So whenever you talk about position of promoter, you have to say promoter is present at the close to the 5' end of the coding strand, and terminator towards the 3' end of the coding strand. So all the references are made with the help of the coding strand. To explain this concept, they have given a hypothetical sequence to us. The promoter terminator flank is structural gene. So the distance between, or the segment of the DNA between promoter and terminator is called as structural gene, and a gene will have two strands. One strand is template, the other is coding. One is the one which is red is from 3 to 5 direction, the other one is from 5 to 3 direction. Its reading is same like RNA, its reading is complementary to RNA. Now the promoter and terminator flank the structural gene in a transcription unit. The promoter is said to be located towards the 5' end of the structural gene, and it is a DNA sequence that provides binding site for the RNA polymerase enzyme, and it is the presence of promoter that also defines the template and coding strand. If promoter is here and this is the 3' end, so this is the template strand. If you put this promoter here and terminator here, then this will become the template strand and this one will become the coding strand. So position of promoter and terminator, if it is switched, then the template strand will become coding and coding strand will become template strand. I hope this point is clear to you. By switching the positions of the promoter with terminator, definition of the coding and template strand would be reversed. Terminator is located towards the 3' end of the transcription. There are additional regulatory sequences that are present upstream and downstream, upstream and downstream, and these will be discussed later.